The focus is going to be on phase 2 proof of concept and dose finding via adaptive design. We'll start with a general overview of adaptive design briefly, and then show some example adaptive design results in the dose finding arena, a quick mention of some regulatory aspects, and then a case study before we open up to questions and comments. The abstract is contained in the slide set. I'm not going to read it, but I call your attention to the list of designs for dose finding that we'll go over today, and that's at the bottom of the page. So we start with that general overview of adaptive designs, and the definition, pharma definition, and appears in regulatory guidances is along the lines of the following, and that is adaptive trial. It uses accumulating data to modify aspects of the trial without undermining the validity and integrity of the trial. By validity, we mean correct statistical inference, like appropriate p-values, controlling type 1 error, appropriate estimates and confidence intervals, and providing convincing scientific results and minimizing statistical bias. And by integrity, we focus on using pre-planned adaptations, not ad hoc. The trial data needs to be maintained confidentially. And then we need to ensure that there's consistency of results between or among the various stages of the trial. And all of this is intended to minimize operational bias. What can we hope to accomplish with adaptive trials? In comparison to traditional fixed sample size designs, we can usually accomplish one of these three while keeping the other two fixed. So we could either decrease development time, decrease sample size costs, or improve precision and quality of the information we collect. Sometimes we can accomplish two of these while keeping the third fixed, but because they counterbalance one another, it's not likely that we could materially accomplish all three with an adaptive design. But nearly always 1 out of 3. Otherwise, it's not worth doing the adaptive design, and sometimes 2 out of 3. The general structure of an adaptive design requires that the trial be conducted in several stages with access to the accumulating data, and nearly always that includes some sort of analysis, sometimes a formal analysis, and that analysis could be on all or part of the data to inform the adaptive decision. And the adaptations, the types of adaptations are changes in the allocation rule, namely the randomization ratios to the various treatment arms. We could modify the sampling rule, how many subjects will be included in the next stage. We could obviously stop the trial for an efficacy conclusion or a futility conclusion or a safety issue. And the decision rule, the decision rules on the conduct of the trial that's not covered by any of the other 3 items could be modified. But again, the modifications need to be pre-planned. So at any stage, the data may be analyzed, and then the next stage is redesigned, taking into account all the available data, but redesigned based on pre-planned guidances. The types of adaptive designs are on this slide. We're probably all familiar with group sequential designs that permit early stopping for efficacy conclusions or futility. In phase 1, there are dose escalation designs to identify the maximum tolerated dose, and the continual reassessment method is a classic example of that. The adaptation is to choose the next dose. In phase 2, there's adaptive dose finding. That's going to be the focus of the remainder of this talk. And the adaptations can occur frequently or infrequently, minimally in a two-stage design with a single interim. And the key adaptive aspect of phase 2 adaptive dose finding trials is to change the randomization fraction or the choice of doses that continue to the next stage. There's blinded and unblinded sample size re-estimation to increase the sample size if results indicate that the planned sample size is inadequate. And then there's population enrichment to focus the randomization in a subpopulation. And then there's combined phase 2 and 3 trial, which is a type of dose selection trial, but in a more confirmatory stage where a dose might be stopped during the trial. Why consider adaptive dose finding trials? Well, they're intended to improve the drug development program efficiency. An ISR report, 2012, indicated that inappropriate dose selection was a key reason for failure in phase 2 and 3. And the aim of adaptive dose finding trials are to focus the randomization on the area of the dose response curve that's of key interest. For example, on the right-hand panel is a sample dose response curve. And if we were in a fixed sample size design, we may choose a low dose, a mid dose, and a high dose. And the low dose and the high dose may not be of key interest because the low dose may have inadequate response and the high dose may have toxicity issues. And we would have rather focused the randomization on the sloping part of the dose response curve. And that's the key aim of adaptive dose finding designs, and you'll see how that occurs as we go through the talk. Another advantage of adaptive dose finding trials is to combine the traditional proof of concept trial and the follow-up dose finding trial. In a traditional phase 2 program, often the highest dose permissible versus placebo is studied in a proof of concept trial. And then when that trial is successful, a dose finding trial is done. And sometimes a definitive dose finding trial, it needs to be conducted to follow up and confirm the dose before you go to phase 3. But in an adaptive trial, with modest increase in sample size, one can accomplish both of those objectives in the same trial. And if you put a futility analysis in, you could, if the drug is not working, you could stop the trial earlier for futility. But then by the end of that trial, with a smaller number of patients than would have been used in the traditional program, you can complete the proof of concept and dose finding objectives with a single trial, and then move to that definitive dose response trial if needed, and phase 3, and that can speed development. An alternative to the traditional definitive dose response study is to conduct your 2 pivotal phase 3 trials with the target dose and 1 dose higher to confirm the target dose, and then the second trial with at the target dose and one dose lower, again, to confirm the target dose. Or you can consider a seamless phase 2-3 adaptive design, an example of which we're going to talk about later. Next, we're going to talk about how to compute power and other performance characteristics for adaptive dose-finding trials. We're all probably familiar with the traditional method of computing power for a non-adaptive trial. They're closed form calculations, and that makes computing power pretty straightforward once we have estimates of the variability and the target treatment effect. But for adaptive dose-finding designs, there are no closed form formula from which to compute sample size. So we need to use simulation, and that's carried out via steps as follows. Obviously, we assume a true underlying treatment effect and standard deviation. Then we generate simulated data for the first interim analysis using those assumed true values. Then we apply the adaptive algorithm to assign doses to the next set of data to be simulated. We repeat steps 2 and 3 until we reach the planned sample size or until we reach our stopping criteria if we insert one. And then we carry out the final analysis on the simulated data and repeat that many times and count the proportion of simulated trials that reject the null hypothesis. And that's the estimate of power for the adaptive design. But we also accumulate additional performance characteristics. And those include the probability of choosing the correct or nearly correct dose, and the number of subjects assigned to the doses with the target level of response. And obviously, number of subjects assigned to all the doses, because we want to check that we minimize randomization to doses that are either too high or too low. And then if we have interim stopping, we obviously summarize the total sample size that has been achieved. So let's now look at an example. This is a frequent adaptation, proof of concept plus dose finding design. I consider either 2 or 3 doses plus placebo. An advantage of adaptive dose finding designs is to include more doses than normally would be included because we're minimizing randomization to doses that are either ineffective or overly toxic. And so the focus of the randomization usually ends up with a small number of doses anyway. So you could include more than this, but I'm just showing the example for the usual type of traditional dose-finding designs that are 2 or 3 doses plus placebo. So we'll consider a total sample size of 102 and 9 sequential cohorts. The first cohort is 30 patients randomized equally to the doses in placebo. And the last 8 cohorts are 9 patients each, 3 to placebo and 6 to one of the doses. And the doses for the subsequent cohorts are assigned adaptively using a standardized difference from a target level of response. The response is a 0 to pain numeric rating scale response. And we simulated the above design 500 times for each of 3 or 4 true underlying dose response curves with an assumed standard deviation of 2.5. The characteristics are averaged over the 500 simulations to compute power and the number of subjects assigned to each dose. The true underlying dose response curves that were simulated are plotted here. The y-axis is reduction in pain, so higher is better. And the dose response curves are numbered 1 to 4 for the 3 dose plus placebo case at the top, and 1 to 3 for the 2-dose case at the bottom. The adaptive design is a T-statistic up and down design. The goal is to find a dose with target level of response, call it R, and the goal of the trial is to assign as many subjects as possible to the dose with that target level of response. The dose assignment rule is based on a T-statistic, the classical T-statistic. That's the observed mean minus the target divided by the estimated standard error. And if the T-statistic is negative, that means that dose has an effect that's below the target, so we increase the dose for the next cohort. If the T-statistic is close to 0, we repeat the dose, and if the T-statistic is positive, we decrease the dose, because that means we have a level of response that's above the target. And so here are the results from the 2-dose case. The power for the first 2 dose response curves was about 93, 94%. A traditional design with equal allocation across the doses would have 90% power, so there's a little bit of a power gain. The type 1 error is controlled. That was the 3rd dose response curve, which was flat. And the number of subjects assigned, or the percent of subjects assigned, is shown in the histogram. And each set of 3 bars at doses 0, placebo, and doses 1 and 2 are in order of the dose response curves 1, 2, 3. So for dose response curve 1, that had a high level of response at the low dose, you could see that more subjects are assigned to the low dose than the high dose. The dose response curve 2, they're roughly similar, and dose response curve 3, which was the null case, minimizes randomization to the low dose and focuses more at the high dose, which is what we want. In the 4-dose case, in the 3-dose case, the advantage of the adaptive design improves. The power for the first 3 dose response curves, 1, 2, 3, was high, 97, 97, 93% by a slope test. And the traditional design now has fewer subjects per group and had power 81, 91, and 89%. I think that 81 is a typo. I think that's closer to 91%. No, it was 81. I'm sorry, that's right. It was 81 because of the plateau type response to the slope test. There's a slight inflation of type 1 error, perhaps. But again, this is phase 2 dose finding, so we could tolerate a modest increase in type 1 error. If we needed to control type 1 error, we could, by simulation, compute a different cutoff for that T-statistic. And in the histogram, now you can notice even further advantage of the adaptive dose finding for dose response curve 1. The randomization is focused on the lowest dose, and for the dose response curves 2 and 3, it's more even. But for the null case, there's very little allocation to doses 1 and 2. We could insert futility analysis. I ran that in this case with a futility analysis after the first cohort at 30 patients and then a second one after 60 patients. And I used an O'Brien-Fleming-like type 2 error spending function and a POC+like function, and the results in the null case for the null case are summarized there. For the O'Brien-Fleming, there was a 15% chance of stopping at the first interim and 41% at the second. So most of the time, the trial would stop early and not have to go to beyond 60 patients. For the Pocock-like spending function, there's a little bit of loss of power, 5 or 6 percentage points, but the probability of stopping early is much higher. The next slide shows a two-stage design. In this simulation, there were 13 on each of 3 doses plus placebo, using the same set of specs as the 3-dose design. And we inserted an interim analysis to drop doses that are likely to be ineffective, those that would have conditional power less than 20%. Then the second stage took the remaining subjects and divided them equally among the doses that were not dropped. The power via pairwise testing was 92%, 85, 82, and 5 for the dose response curves 1, 2, 3, 4, respectively. And it was 89, 92, and 88 via the slope test. But the key point is to look at the doses, the percent of simulations that each dose was not chosen for the second stage. And you can see for dose response curve 1, there's very little chance that the doses would not be selected. For dose response curve 2, there's a much higher percentage chance of dose 1 not being selected, and that's good because dose response curve, the green dose response curve, has a low response for dose 1, and then the probability of being dropped is higher for dose response curve 3 for the 2 low doses, which is what you want. And there's a high probability of not taking any doses to the second stage, 60% for the null case. So you could see the advantage of even a single interim analysis when you drop doses. Another example of a adaptive single interim analysis dose-finding design was used for a client that we worked with. And I just present the layout of that to give you an idea of the type of options that exist. In this trial, 40 or 50% of the total sample size was randomized in equal proportion to 5 treatments, as shown in the matrix below. The 0 dose, which is placebo, and then once-a-day doses 0.2, 1, and 5 milligrams. And then we wanted to also study a twice-a-day dose. And so there was a twice-a-day dose. And then once we got the results of stage 1, the options for stage 2 were to treat the remaining subjects among doses that could be chosen from the list shown in the matrix under the second stage. So we could pick middle doses, or we could pick the original doses from stage 1, or we could expand the BID to lower or higher doses. Now, naturally, we wouldn't choose all of these doses that are listed. They were just pre-specified to be chosen. You could also consider an option to add a third stage, but this example didn't have that. And then on the next slide, there was an alternative approach where we would have a pilot phase, or the stage 1 would study 6 active treatments and a placebo, 2 BID regimens and 3 once-a-day regimens, and then any of those could have been moved to the second stage. The analysis of this will be talked about later. Now we'll move to some other types of adaptive dose finding methods. The first is a Bayesian algorithm that was developed in collaboration with Scott Berry and implemented in software that we have in-house. The idea is to use Bayesian updates of model parameters after each cohort. We assumed an S-shaped 4-parameter logistic model and treated the 4 model parameters as random variables with a specified prior. After each cohort's response, the posterior distribution of the parameters are updated and the models re-estimated. The algorithm uses a minimum weighted variance utility function that I'll show you later for decision-making during the adaptations, namely the randomization ratios are proportional to the weighted variance function at each dose. And they're chosen by the function that minimizes the weighted variance. The reason we use a 4-parameter logistic function is because it's very versatile. This slide shows several examples of dose response curves of different shapes that can be configured with this 4-parameter logistic model. So it's very versatile because it can even fit a linear or a gently sloping line or an exponential type line or an S-shaped line. Here's the function again. We assume normally distributed random error and added onto the 4-parameter logistic function. The patients are randomized in cohorts. Within each cohort, there's a fixed fraction allocated to placebo, and the remaining subjects are assigned adaptively in proportion to the weights that minimize the weighted variance function in the lower left. Another type of Bayesian adaptive design is a normal dynamic linear model, or NDLM for short. It's a Bayesian forecaster, and it uses a parametric model with parameters that are updated after each cohort. And the forecast is derived as probability distributions and provides facility for incorporating expert information. The NDLM idea is basically a smoother to the estimated unobserved dose response curve. And I'll show you that on the next few slides. So if we set up the data structure to let theta sub I or theta sub t to indicate the mean response at each dose for doses 1, 2 up to some number of doses, and 0 dose as placebo, then obviously the aim is to estimate the mean response vector. And the model that's used is on this slide. It's very straightforward. The observation, the response y, given the value of theta, is a linear function with an error that's assumed normally distributed. And then the mean at dose t, given the mean at the previous dose, is also linear with a different, with a normal distribution and a different variance parameter. And so the variance parameters are assumed to be independent, and we only need a prior on theta 0 because all the other thetas are functions of theta 0 according to the structure above. So we assume a prior on the placebo response, and usually we do have prior information on placebo, so we can make an informed prior, and then we have the sequence of unobserved state vectors theta and the values y, and so we can update this after each cohort and get a better allocation of subjects to optimize the estimation of these model parameters. It kind of functions like fitting a slope at each point on the dose response curve. And this slide shows how the model works for samples of 200 subjects spread across 6 doses, 5 doses in placebo. So in the upper left, there's a flat dose response curve, and there's a sequence of repeated sampling from this data, and the gray area shows all the individual outcomes with I think that's 95% bands around it, and the dotted the dashed line, the solid black dashed line are the averages. And so you can see for each of the types of dose response curve, this normal dynamic model does a really good job at estimating the true underlying dose response curve regardless of shape. The last or second to last type of design I'll talk about is the maximizing design. And this design is for an umbrella-shaped dose response curve or a unimodal dose response curve. Note that the NDLM design could be used in this circumstance as well, because for that quadratic design, it can model a unimodal dose response. So, and then another design is this maximizing design for such an umbrella response. And an example of an umbrella type or unimodal response is a composite utility function combining safety and efficacy. For example, some weight times the efficacy response plus a weight times a safety response. And the objective is to maximize the number of subjects assigned to the dose with the highest mean utility. And the aim is to improve power for discriminating the dose with the peak response from placebo. Obviously, the assumption here is unimodal dose response. And the proposed methodology is based on an old algorithm that was implemented by Anastasia Ivanova and published in 2008. And the way that works is as follows. Each cohort is assigned an adjacent pair of doses. For example, if the current cohort is at dose 3 and 4, and there's a negative slope, then the next cohort would get 2 doses lower because a negative slope means the peak is to the left. And if there were a positive slope, then the peak would be to the right. So we would increase the dose for the next cohort. But if the slope is relatively flat, then we'll repeat the dose. There will be an example of a case study used in maximizing design at the end. The last type of design is a 2-stage design, which we talked about earlier, but the way it works is what we're going to talk about now. There's a fixed total sample size and a certain number of treatment arms, including placebo. And in stage 1, the subjects are equally allocated to all the arms. And there's analysis after stage 1 to choose the best treatment arm or the best several treatment arms. And then at the second stage, the remainder of subjects are equally allocated to the selected arms and placebo. And the final inference is made combining the results from both stages via pre-specified sum of weighted Z's. And this weighted Z methodology controls type 1 error in the strong sense. This approach is very flexible. Several functions and methods for multiplicity adjustments are available, and they permit data-dependent changes to adapt sample size, to drop arms, and you can take several arms to the second stage. And most importantly, it's not necessary to pre-specify an adaptation rule from the statistical point of view. But it's obviously necessary to have some kind of guidance on how to select the dose. But the data monitoring committee can look at the data of the primary endpoint plus important secondary endpoints and consider safety together without a pre-specified utility function, but with the aim of picking the dose that overall gives the best efficacy signal after a clinical review of the efficacy data and has acceptable safety profile from a clinical review of the safety. And then implementing the weighted Z analysis methodology with appropriate multiplicity adjustments strongly controls type 1 error. And that's what I just said on this slide. So now a brief slide on regulatory aspects. The FDA has a guidance on adaptive designs, and it classifies them into adequate and well-controlled trials or adequate and well-understood adaptive trials. And those are trials for which the statistical properties are well-known and analytically verifiable from a mathematical point of view. And those are designs that can be used for pivotal trials. There are less well understood designs that the agency needs to gain more experience with. And the types of adaptive dose-finding designs that we're talking about here, except for the two-stage design with the weighted Z methodology, but those frequent adaptation type designs like the T-statistic design, the Bayesian 4-parameter logistic design, the NDLM, the maximizing designs are probably less well understood, but that doesn't mean they're unacceptable. The guidance encourages their use, and because they're used in phases 1 and 2, there's less stringency about them. The adaptive designs are reviewed by the agency in the context of the overall submission package, namely the development plan. And if they're in the learning phase, then there's more leniency with regard to type 1 error control and documentation of type 1 error control, but not in the confirmatory phase. If the trial is viewed in the confirmatory phase, then obviously it would be viewed with greater stringency. So the adaptive trials must make sense and add value to the development plan. And as mentioned, there are fewer possibilities for adaptation in confirmatory trials. The agency encourages discussion about the design, proposed adaptive designs prior to carrying them out. They obviously look for evidence of control of type 1 error. And the other key aspect are, especially in confirmatory trials, to make sure that the integrity of the trial is maintained, and there's a need for firewalls and evidence that firewalls are maintained during the trial to protect and keep blinded the interim analysis data from all those except the data monitoring committee. So we'll close with this case study, and then we'll have time for questions and discussions. The case study is a frequent adaptation maximizing design. The next couple of slides are just a quick overview. There's a more complete presentation at the link here, and you were given the link to download this slide set, so you'll be able to get this link if you're interested to see the complete slide set. And then these three references document the trial. The first reference is the original maximizing design reference. The second reference is the design itself and the simulation report that was justified the design. And then the bottom reference is the reference that reports the results of the trial. So here's an overall summary of the trial, the design and the outcome. It was a phase 2 trial, placebo-controlled and active-controlled, and it was post-bunionectomy surgery. The objectives were proof of concept and estimating the dose regimen with the optimal balance between maximizing efficacy and minimizing intolerance. The active control was morphine in the post-bunionectomy, first 48 hours post-bunionectomy. And so morphine is highly effective, but has important tolerability issues, namely mostly nausea and vomiting. So the aim for this treatment was to compare against morphine and benchmark against morphine. The maximizing design was chosen to yield better quality information than just a non-adaptive equal allocation design, because fewer patients would be assigned to dose regimens which are either ineffective or intolerable. So this was a good fit for this type of design. The true potential efficacy and tolerability dose response curves were constructed separately for efficacy and tolerability to span a range of potential true underlying dose response curves. And then several options for a clinical utility function were considered to combine them. There's more about that in the slide set, the link of which was on the previous slide. But briefly, we started with a weighted function like on the maximizing design slide, but the clinical monitor with whom I worked on the design didn't like that, and so he came up with his own utility function, which I'm going to show you on the next slide. And so then we computed the utility functions according to his specification, using his specification to combine the efficacy and tolerability curves. And then we simulated and reported the performance characteristics, and the trial was carried out. And the results indicated that the maximizing design had high probability to estimate the correct or nearly correct dose with maximum utility, and it maximized the assignment of subjects to the target dose, and it minimized the assignment of subject to doses away from the target dose. So the sponsor really liked that, and we implemented it. The way the study was actually carried out was in two stages. There was a proof of concept stage and dose response seeding stage. So the initial cohort was 150 patients randomized in equal proportions to placebo, active control, or 4 test drug regimens, actually. And then the enrollment was paused for a month while we analyzed that stage 1 data. And then the second stage was the maximizing design. There were 10 successive weekly cohorts of approximately 25 patients planned, and that depended on a weekly enrollment rate. The trial was actually carried out with treatment of all the patients on Monday. There was 48-hour observation period. The data was collected electronically and available by the end of Tuesday. So that Monday morning, the Independent Stats Center had the data and had Wednesday and Thursday to run the analysis tables and then met with the DMC on Friday. And then the DMC met on Friday, reviewed the data, and reviewed the maximizing design recommendation, and then sent the 2 doses to the IWRS by the end of Friday, and it was implemented for the next cohort the next Monday. Each successive cohort of those 25 patients was randomized in 4:8:5 ratio to placebo, the 2 doses of test drug, and morphine. And the expected yield from the simulation was 65 placebo patients, 75 active control patients, and 80 to 100 patients on the dose with target level of response. Sorry for the typo there. There's some extra information, but that's what the simulations yielded. And here's the utility function. The columns indicate varying degrees of tolerability. T is test treatment, AC is active control, and so the first column there is tolerability better than the active control, namely the difference in adverse experience rate and the adverse experiences of interest were nausea and vomiting. So if there was a 20 percentage point advantage, that was considered better tolerability than morphine. If the difference was between -20 and 0, then that was considered a little bit better. If it was between 0 and 20, that was considered a little bit worse. And more than 20 percentage point difference above morphine was considered worse than morphine. Similarly, down the rows, the efficacy was, and this is difference on a 0 to 10 numeric rating scale. if the pain intensity reduction difference was bigger than 1 unit worse than morphine, that was considered less efficacy. If it was between 0.5 and if it was within 0.5, that was considered similar efficacy. If it was between 0.5 and 1.5, that was considered better. And if it was a difference greater than 1.5, that was considered much better. Now you notice the gap between the less efficacy and similar efficacy, and that was okay because what we did was we assumed that these values were the midpoints of those ranges, and then we used two-dimensional linear interpolation to get the actual estimated utility from this table for each of the observed differences in tolerability and efficacy. This table was laid out and achieved in discussion with the clinical monitor. And then the trial was carried out, and here are the results for efficacy. This is pain intensity, mean pain intensity plus or minus a standard error. It's copied from the results paper. The gray is placebo. The yellow or gold is morphine. And so you can see the morphine effect on pain intensity. It's a reduction. Excuse me. And then the increasing shades of blue are the increasing doses of the test treatment. So you can see strong evidence of a dose response, and you can see that the top two doses look like an advantage to morphine, and the lowest dose and the lowest two doses showed kind of similar to morphine early on, and then overall generally similar to morphine later on. And so obviously the treatment's working. And then here's a summary of the tolerability data. Dizziness and headache are there just for completeness. The primary utility was computed by proportion of patients with nausea or vomiting. And so if you just look at the nausea row, you could see on the right, morphine had 56% incidence, and the high dose of test treatment was higher, but generally similar at the 2 milligram dose and lower at the 1 milligram dose. And that relationship is generally similar for vomiting. And so the results of the trial indicate that the 3 milligram dose had better efficacy than morphine, but at the cost of worse tolerability. The 2 milligram dose had better efficacy than morphine with similar tolerability. So there's an efficacy advantage with similar tolerability. The 1 milligram dose had better tolerability, but generally similar efficacy to morphine. And so this information, when submitted to the regulatory agency, yielded breakthrough therapy designation for the test treatment for the management of moderate to severe acute pain. The NDA was filed last January. The advisory committee voted 8 against and 7 in favor. And the reason was inadequate safety information from phase 3. And so they requested more safety information. And the reason for the key reason is because, you know, morphine is a controlled substance because of addiction, and they wanted more evidence with regard to the potential addictive effect of this treatment. And so that safety information is being assembled and pending. And for more information, you go to the Trevena website. The link is there. This slide has a reference list, and then we get to the end of the first hour, and we can move to the question/discussion section.
Thanks, Jim. Hi, audience. This is Mike Weitz, and I'm the Director of Marketing here at Cytel, one of the directors. So we'd like to move into the question and answer period right now, and we're looking at many questions that are coming in through the Q&A, and we'll address as many as we can with the time that allows. Before we begin, however, let me first just thank Jim for his prepared remarks, and to let you know that we'll also be joined, Jim will be joined by colleagues currently in France, Benjamin Espeni, and together with Christelle Pommet, and so they will be assisting Jim in responses and helping to select your questions. Once again, everyone on the webinar right now can download the slides simply from cytel.com/events, and we'll keep that link open through until Monday, at which point the free access to the slides will conclude. We'll also let everyone know when today's recording is available for replay, and that link will continue on for those who registered for today's presentation. So let us begin. We have heard, while we're looking at the questions and continuing to assess them, there are questions that that Jim and the team have faced in the dose-finding arena with adaptive dose-finding designs. So Jim, and colleagues, let's begin with, is there any particular method that you'd favor, or perhaps even to avoid, if a non-monotonous dose-response relationship is expected?
Yeah. The T-statistic design and the 4-parameter logistic design need the assumption of a monotonic dose response. So obviously you want to avoid them in that situation. For the non-monotonic design, if it's a unimodal design, then either the maximizing design or the NDLM is useful. The difference would be that the maximizing design would likely do better at allocating subjects to the dose with the higher, with the peak utility. The NDLM tries to fit the entire curve, and therefore it would spread the observations more across the range because it's trying to fit the entire curve, and therefore it would still do a good job at identifying the dose with maximum utility, but it wouldn't put the most subjects there because it's trying to model the whole curve. So it depends on what the objective is, and you obviously have to simulate both of them and compare their performance characteristics in order to pick one for each for the set of true underlying curves that you're considering. And then depending on what your objectives are and the priority of your objectives, if your objective is to get a model of the curve and estimates at different places along the curve, then the NDLM is probably better. But if your objective is to put the most subjects possible at the dose with best utility, then the maximizing design is probably better. So in summary, avoid T, avoid 4-parameter logistic, and focus on NDLM and maximizing.
And Jim, before we continue with some more detailed questions about the statistical issues, let's consider an operational issue. It's not always easy, and in fact, often disruptive, to press pause once an enrollment has begun. So what are your thoughts, and do you have on the adaptive approaching and considering adaptive decisions while a recruitment is ongoing?
Yeah, that's an important point to consider. In the two-stage design, enrollment often continues using the original randomization list. And depending on the timing of the duration of treatment before you get the primary endpoint readout, you could have considerable over-enrollment in the trial by the time you get the interim analysis results for the first, say, half of the trial, if that's when the interim is. And so the logistics there, if you have a long time to observe response during treatment, contraindicate the use of an adaptive design. And that's an important logistical aspect to keep in mind. The example that I talked about was the case study was a 48-hour window, and it was easy to just keep the enrollment going and get the interim results in time. In a two-stage design, though, with a considerable sample size, it's not too bad to have a pause and have two sections of the trial. But again, you have to balance the time that it takes to do that versus the logistical aspects of stopping and starting. But it's an important point to consider, and you have to do that work and talk to the operations folks because you need to work that out. And the simulations can be done with the software that we have. The simulations can be done to account for that lag in the information and the over-enrollment in the first stage or the earlier stages.
Jim, I'd say that serves as a reminder that adaptive design is one thing and successful implementation is quite another.
Right. No, that's right.
Yeah, I would, especially in later stage trials, confirmatory trials. So Jim, and Alexander Ooms asks some specifics on your dynamic linear model slide. Alexander wanted to know what type of functions do FT and GT represent. Is that something that you could go back to and address?
I tried. Yeah, there you go.
Hi, Jim. I think it was slide 28.
Yeah.
Here. Yeah.
There
here.
you go. No, capital FT and capital GT are parameters. They're a coefficient for theta T. That's not a function. That's a parameter.
Thank you, Jim.
And Jim, we have a Bohemia Bittman, I believe, is the forgive my pronunciation. But they want to know if are these methods included available in today's EAST software?
No, not in East. Cytel has a different tool called Compass. And Compass has not only the designs that I talked about today, it has an Emacs model design as well. And these designs are in Compass for binary endpoint or for a continuous endpoint. The two-stage design is in East, but the other four designs are in Compass.
Thanks for clarifying.
They may come to East in the future, but it's not on the near-term priority list because they're available in Compass.
And Jim, can you confirm that Compass can permit to do simulations as well?
Yeah, Compass carries out the simulations and provides summaries. Those plots are the dose response curves, and those histograms that I presented, those were copied out of Compass. But then Compass also has the execution stage where it records every adaptation, the data that was used for every adaptation, and every interaction with the tool during the conduct of the trial. All of that gets recorded for documentation purposes. So the Compass tool does both design, simulation, and the execution.
Thank you, Benjamin and Jim. So Andrei Pavlov poses the question, and this is something that occurred to me too, listening. For the less well understood adaptive designs, as the regulators refer to them, what type of documentation do regulators expect to support the validity of the proposed design? What's the documentation for?
Yeah,
maybe it lies in simulations as well.
Yeah, there's a simulation report that is written and attached to the protocol as an appendix, and that simulation report goes into detail to identify the methodology that's used, the true underlying dose response curves, the adaptation rules, the specs for those rules, and then the simulation results and summary tables of those simulations. An example of the content of that would be the 2015 reference on this slide that I'm showing on slide 38. Well, the number's 38 there. It's the references slide to the case study, the Therapeutic Innovation and Regulatory Science 2015. That publication would be an example of the type of information that would be in a simulation report.
And Jim, maybe a little further on the less well understood designs. You mentioned about the need, the benefits of communicating with the FDA or EMA when considering the so-called less well understood approaches. But do you have any insights, maybe your own experience, you might want to share with the audience in just those interactions, and that might be insightful or what you might think typical?
I actually haven't received any. I've done maybe half a dozen adaptive dose finding designs in the phase 2 arena, and they were not pivotal trials. And there really wasn't any FDA feedback on them. Obviously, they were submitted to FDA. We didn't get comments back because it was understood that they were in the learning phase. So, but on other occasions, the intention of trials was to be pivotal. And those were adaptive designs, but not dose finding. There was a two-stage dose finding design that we went to FDA with, and the FDA comment was, are you still in the learning stage? And they questioned the use of the design as a pivotal trial, but it was a two-stage design, and the methodology was straightforward with regard to control of type 1 error. But their comment was, you really haven't had proof of concept yet, and you're doing dose finding and proof of concept in a two-stage design, and you want it to be pivotal. And so they suggested that obviously if the results were robust, it might be used as a pivotal trial, but up front they wouldn't commit to accepting it as a pivotal trial. So that went back to that comment where the context of the adaptive design in the drug development project is what they look at. And so are you in the learning phase or are you in the confirmatory phase? And if you're still in the learning phase, it's not a given that the trial could be used as pivotal. But again, it depends on the context of the disease. You know, are there available therapies? Is it a life-threatening condition? Is it a rare disease? Things like that. So all of those factors could come into the discussion as well.
Thanks, Jim. I guess communication is the key.
Right.
So, and Jim, we're going to get back to the questions from the audience, some specific questions from the folks who are so good to stay on with us. And once again, thank you to everyone who's joined us, and especially those who are staying with us for this particular session. So Jim, George Kai, if I'm pronouncing it correctly, posed the question about your thoughts on how best to balance the times of the occurrences of interim analyses, considering the variability of nuisance parameters. For example, a variance in the adaptive design.
So obviously, if you take a total sample size for 80 or 90% power and you divide it by 10, and you do 9 interim analyses and a final analysis, the variability of the estimates for the first couple of cohorts is high, but the thing is, over the long run, in general, you migrate towards the doses with the target of interest. And if you go in the wrong direction after cohort 1, then when you get to cohort 2 and 3, you will achieve a correction. But if you end up going in the right direction, then you're better off than if you didn't do that interim analysis. And so, I mean, that's why you do the simulation. So to answer that question, I've done similar, I've done simulations for some examples where, let's just say you have 100 subjects and you do cohorts of 10 versus if you do the first interim at 50 and then you do cohorts of 10. You get about the same thing, because by the time you get to 50, you've corrected, and by the time you, but in some circumstances you do better than if you wait till 50 to adapt, because you moved in the right direction. And so when you look at those simulations, depending on what the true underlying dose response curves, you have to simulate it for your cases to answer the question. Because you can invent cases where it's better to wait to 50, and you can invent cases where it's better to start the interim analyses at 10. But back to the logistical issue, it might be harder to do analyses after every 10. And so, you might be better served to wait for 50 and then just consider one additional interim and not 4 additional interims. So it depends on the situation. It depends on the logistics, your recruitment rate, how long you have to wait for the observation, and how long it takes to get the data and do the analyses. All those logistics have to be worked out. And then for the cases that you want to evaluate, you need to do the simulations to compare them to answer the question for your case.
Thanks, Jim. Let's move on to Eric Mortensen. So Eric poses a question. Jim, I'm interested in the use of composite efficacy and safety parameters. He says that we considered this approach for one program, but lacking familiarity, it was not adopted. So in your experience, how comfortable are regulators at the EMA and FDA reviewers with the acceptance of such an approach to predicting the dose selection?
Well, it starts with the definition of the composite endpoint and the acceptability of that composite endpoint. But that's a different question. The acceptability of a composite endpoint is a different question than applying an adaptive algorithm to it. Given an accepted composite endpoint, then the issues with regard to adaptation are the same as they would be to a non-composite endpoint. And so I see that as two separate issues.
Fair enough. Jim, Anqing Zhang asks, do you always have the prior information to use, to consider and to incorporate? If no priors are available, what do you consider as an alternate or a substitute?
If we're talking about prior for the Bayesian designs, then you can use an uninformative or a vague prior. If we're talking about how to define the true underlying dose response curves to simulate, if there's no prior information, the scientist you're working with will obviously say, I want to examine this dose range. And then you can write a dose response curve that goes up quickly at the first dose and levels off. That would be the best case scenario. And the worst case scenario is a dose response curve that stays flat and then goes up at the highest dose. And then you can pick 2 or 3 dose response curves in between. And then that's a spectrum of possible curves. And then you can discuss with a clinician, okay, here's a whole set, best case, worst case, and several in between. Could it be any of those? And he says, yeah, it could be any of those. So then you want to simulate all of those. And he said, and then, the discussion could focus to certain ones to give priority to, and then those you would give priority to. But it's an interactive discussion with the clinical person to come up with a set of curves that span the range of potential curves that you want the design to work well for.
And Jim, we're going to go back to the classical T-statistics method that you mentioned earlier. The question is there any particular reason for choosing minus 0.1 and plus 0.1 as the values, your threshold values?
Yeah, that's a good question. And I actually worked on this design with Anastasia, and she says the reason we pick 0.1, which is arbitrary, that value is arbitrary, but the smaller it is, then the more chance there is to bounce around. And you want the design to bounce around. You don't want it to get stuck. And so making it small is why, that's why it's small. I've run simulations using 1 instead of 0.1. And it does get stuck sometimes at a dose that's close, but not the right one. And so that's why it's 0.1. But it is arbitrary. And again, it's good to do at least some sensitivity analysis in your simulations for the cases that you're studying, to maybe vary that. And when I say vary that, I don't mean in a continuous way. I mean, like, you might consider 0.1, you might consider 0.5, you might consider 1. Sometimes I've considered 2 even, but that's too extreme probably. The other point to consider is that Compass has a facility to split the active patients with a T-statistic for a target 1, a low target, and a target 2. So instead of assigning all the subjects to one target, it splits them in half and assigns half them to a target that might be minimum clinically important difference, and then the second target might be twice the minimum clinically important difference. And then you get the randomization clustered around 2 places in the dose response curve. And Compass accommodates that. That's a kind of an extension of this design.
Thanks, Jim. Andrei Pavlov has another question that's interesting. He asked, Jim, could you elaborate on how the optimal power is defined in the multiple dose setting? So what alternatives are considered, please?
Yeah. Typically, it's either the mean that's estimated from the fitted dose response curve versus placebo at each dose. And in the monotonic situation, you would obviously do a step-down testing. The data from the T-statistic design, which is not a model-based design, but that data is summarized via isotonic regression. And the estimated means during the trial are derived via isotonic regression. So you can do step-down testing based on the isotonic regression means. The alternative is, or in addition to that, you could do a slope test, a trend test. But there's a design that we did not talk about, and that's MCP mod design. That stands for multiple comparison modeling design. And that design uses several potential underlying models and then does multiple comparison adjustment to control type 1 error to test for treatment effect. That design is in EAST, but only for non-adaptive. And there aren't many examples of that I've seen in using that design in an adaptive sense. But the answer to the question is it's either mean from the fitted model versus placebo, or via step down, or a trend test.
Thanks, Jim. Benjamin speaking. I have a last question still related to the T-statistic up and down design. So on the slide you are currently presenting, it seems that in that design, the targeted mean response play a key role. And I'm wondering if there is any risk to miss some doses that can give better response rate than the target? And also, is it possible to revise that value during the course of the trial?
The answer to the first question is to use 2 targets and split the sample size to be assigned to the dose that has, according to this T rule, with an R1 and an R2. Obviously picking R1 and R2 leaves the same question, and Compass has ability to do R1 and R2, but it doesn't have ability to do R3. But then the more targets you use, you would include, the less sample size would be available for each one of them. And so if you have more targets, then you're probably better off considering like the 4-parameter logistic design or the NDLM design, which models the whole curve. The second question was, tell me the second question again.
It was, is it possible to revise that value?
Oh, revise, yeah. No, I don't know. You'd have to have a rule for revision. And you could envision a rule for revision, something like we hit a dose that has X% conditional, not X, right? X% posterior probability that the response exceeds a certain value. And once you hit that, then you change the target to 2R or something like that, or 1.5R, or, you could pre-specify it. So you could envision pre-specifying some rules for that and simulating it. But you would have to write your own software to simulate that. It's not a hard program to write because it's an easy type of decision rules. And so what you just said, Benjamin, is the potential for an improvement on this design that you could put in your research list.
I see. Thanks a lot, Jim.
And at this point, I too will thank Jim, especially, and Benjamin, Christelle, all here at Cytel, and extend on their behalf thanks to all of you who joined us and shared your questions, your interests. That's very encouraging to us here at Cytel to see such interaction and such interest in design innovations and their successful implementation.